Hello and welcome to another edition of Zog Science. It's been a little bit, but we did the last unit without videos. So this next unit we're going to be starting is the cell cycle, and we're going to begin by talking about mitosis. So there is a Latin phrase, omnis cellula e cellula, which means every cell from a cell, and that is basically the kind of guiding principle for us here. All cells come from other cells. Now, obviously, this brings up the question, where did the first cell come from? And we'll discuss that more when we get to our evolution unit. But for now, we're just going to go with every cell comes from another cell, and we call this cell division. And cells go through a cell cycle, much like animals and plants and other organisms go through a life cycle. We're going to be talking about the cell cycle. So where we start is where we're going to finish. Now, the life of a cell goes from a parent to a daughter cell. When we talk about these terms, we're not really implying gender. We're just saying that we've got our starting cells and then we've got our daughter cells. Uses are for reproduction in single-celled organisms like bacteria, protists, things like that, as well as growth and repair in multicellular organisms, such as us. Now let's talk about how kind of genetic stuff is organized. We've got our genome, which is the total amount of information stored in the DNA. We've got chromatin, which is DNA and proteins found in a cell, and then you've got chromosomes, which is just the DNA structure. We're going to be using these terms a lot, which is why we want to kind of talk about them. Now, each species has a unique number of chromosomes in their cells. You've got two different types of cells for multicellular organisms. We've got somatic or body cells, which contain two sets of chromosomes. We annotate that as 2N. For humans, that number is 46. For gametes, which are your sex cells, they contain a single set of chromosomes, which we would annotate as N, and in humans, that is 23. So you can see that 2N, 2 times 23 is 46, all right? And we'll get into more about the chromosomes and kind of how that works in a later video. Now, chromosomes during replication, normally they're uncondensed in a long fiber. So what that means is that they're kind of spread out. You can't actually see them underneath a microscope. However, after replication has occurred within the cell cycle, they're going to condense to a shorter, thicker form where we can actually see them. And when this happens, replication has occurred, so we've got a pair of chromosomes, which we call sister chromatids, that are genetically identical to one another as long as there haven't been any mutations. And they are held together by the centromere, which is right here kind of in the center. And notice that it kind of produces this X shape because there's a little bit of pinching that occurs at the centromere. And what's going on at the centromere is that you've got similar sequences of DNA that help stick to one another, as well as a protein complex. There are two stages of the cell cycle, not two stages of the cell cycle, two stages of cell division, which are mitosis, which is the division of the nucleus, division of the genetic material, and then cytokinesis, which is the division of the cytoplasm, all the other organelles and things like that. Phases of the cell cycle. So in general, we've got 4 phases to the cell cycle. We've got G1, which is growth, S, which is DNA replication or DNA synthesis, G2, which is some more growth and preparation for mitosis. And a human cell on average divides about every 24 hours. Some cells divide more often, some cells divide less often. But this is just kind of a general guiding principle. Your G1 phase is about 5 to 6 hours. S phase is 10 to 12 hours. So DNA replication takes a little bit of time. That's because you want to, there's a lot of DNA to replicate, but also you've got some fact checking, proofreading that goes on. And then we've got G2 phase, which is 4 to 5 hours, and then mitosis, which is less than 1 hour. So this is kind of all of the stages of mitosis and as well as cytokinesis kind of tacked onto the end. You have learned this before, I'm sure, in your freshman biology class. We've got the acronym IPMAP, except now we've added a second P. So it used to be that you all just learned interphase, prophase, metaphase, anaphase, and telophase, and then cytokinesis at the end. Now we're going to add on prometaphase as kind of a intermediate step between prophase and metaphase. So G2 of interphase, at this point, the nuclear membrane and the nucleolus are present. We can see them. Our centrosomes are going to form from a centriole pair. So a centrosome is kind of the whole thing with centriole pairs being inside. And what's going to happen is that the cell is going to be preparing. There's some regulatory things that happen, and we'll discuss the regulatory things in the next unit. Then we've got prophase. Prophase involves the chromatin condensing into chromosomes. So now we actually begin to be able to see the actual chromosomes. The nucleolus is going to disappear. Chromosomes are going to appear as the sister chromatids. Remember, so they've got kind of that X shape. And the mitotic spindle is going to begin to form. The mitotic spindle is a structure that is going to help guide our chromosomes and move them from to the opposite ends of the cell. And the centrosomes are going to begin to move to the opposite ends of the cell, or what we're going to call opposite poles of the cell. Similar to kind of the north and south pole of the Earth. This is a little bit closer in-depth look at the mitotic spindle. So what we've got going on is these what we're going to call kinetochores. I'm sorry, not kinetochores, microtubules, which are connecting between the two centrosomes. We've got some of them that are going to connect to the actual chromosomes, and they connect at what is called the kinetochore, which is right around the centrosome of the chromosomes. And then you've got some microtubules that are going to connect with other microtubules from the opposite pole. And they're going to be connecting right along the metaphase plate, which we're going to see here in just a minute. So in prometaphase, the nuclear envelope begins to disappear. Or disappears completely, I guess. The spindle connects to centrosomes at the kinetochores, so we're lining up, or they've attached at this point. And again, those microtubules from one pole are going to be attaching to microtubules from the other poles as well. In metaphase, the centrosomes have moved to completely opposite poles of the cell. Our chromosomes move to the metaphase plate. That's an imaginary kind of plane right along the middle of the cell. And the kinetochores of each sister chromatid are attached to the microtubules from the opposite poles. So they're right there along the end. Kinetochore for one sister chromatid and the one from the other are attached to opposite poles. During anaphase, this is the shortest phase. The sister chromatids are cleaved apart, so an enzyme is activated that breaks the kinetochores, breaks the centrosomes. The kinetochores of the microtubules shorten, so when they do that, they pull the chromosomes toward each pole. So the ones that are attached to the actual chromosomes shorten. The ones that are attached to non-kinetochore microtubules, or the ones that are attached to opposite poles, will elongate, so they begin to push the cell apart while they are pulling the chromosomes in. And by the end, each pole of the cell has a complete set of chromosomes. In telophase, the daughter nuclei begin to form. The nucleolus, or the nucleoli, which is the plural for that, begin to reappear. The chromosomes are going to uncondense, so they begin to unravel again. Microtubules are broken down because they're no longer needed, and mitosis is complete. In cytokinesis, we get a pinching that occurs, which we call the cleavage furrow, that begins to bring the cell into two, splits the cytoplasm apart. All of the organelles have moved to opposite ends of the cell at this time as well. So each cell will have all the things it needs to perform its functions. Now, some differences in plants and animals. Plants do not have centrioles, so they line up their, they use a different mechanism for lining up their chromosomes in the middle, which we're not going to learn about. And we've got because they have cell walls, they have a plate that forms kind of in the middle, and those plates then eventually connect with one another, forming a new cell wall, which then splits into two daughter cells, as opposed to the cell membranes just simply pinching in when we're talking about the animal cells. Got a short little video of what it looks like for mitosis to be occurring. So see, at this point, we can't really see the chromosomes. Chromosomes are going to begin to condense, so we are now in prophase. You can start to see them actually appear. Moving into prometaphase. Now we're moving into metaphase. Chromosomes are beginning to line up along the metaphase plate right here. They've lined up. We're going to go into anaphase here briefly. Oh, there's another chromosome that needed to move. There we go. There's anaphase. They're pulling apart. And there is the classic pinching in of cytokinesis. Notice how they kind of fall apart. The chromosomes begin to uncondense. The nucleoli reappear. And that is your cell cycle. Okay, now a little different bacteria. So what we were just looking at was animal cells. So bacteria, they have circular DNA, so they don't actually have linear DNA. So what that means is that there is a place on the DNA known as an origin. The origin is replicated first. The origins then move apart to opposite ends of the cell. And when they do that, that kind of triggers the movement of the rest of the DNA to those opposite ends of the cell. And the new membrane and cell walls are created here. So they're kind of the pinching in again, but now you have to build a cell wall as well. And then eventually that leads to two cells. It's not fully understood exactly how bacteria accomplish division. They haven't really figured out all of the proteins and all those types of things that are involved the way they have with animal and plant division. The reason why we call it binary fission is that it is not mitosis. It's not exactly mitosis because there is no nucleus that actually gets split apart. And I believe that is all I've got for you today. Thanks for watching, and I'll see you next time.